everybody, it's Miss Snook, your science teacher, and today it is now 2 days later during our human digestion laboratory, and it is time for me to look at my results. So I've already entered my special code into the virtual lab, and I clicked on enter code, and it brought me to where I pick my glove, and now I am at my lab bench to look at the results. First thing I'm going to do is I'm going to remove the stoppers from the test tubes on my milk experiment. And we're going to test to see whether or not glucose is present. If you remember from reading the lab, glucose will be present after lactase has interacted with it to separate the sugar from the milk. And so we're going to take our test strip, dip it into our milk. You may or may not see a change in color, but just go ahead and place it on your drying area on the chart for glucose near its appropriate color. And in order to make sure that you are doing this part correctly, you need to click your mouse each time you place your strip in your test tube. Alright, so we'll go ahead and put our forceps down on the table. And let's go take a look at our results. So the first two test tubes tested blue, and this is in this range of the chart, and that says glucose is absent. And then the next two test strips tested orange, and that's on this side of the chart, which says glucose is present. So in your lab report, here, you're going to go down to this chart for the milk digestion data. We've already filled out our prediction, and now you can fill out your test strip results and answer the question, was the milk digested? For test tubes 1, 2, 3, and 4. Next, we're going to go back to our virtual lab and take a look at our egg. Your results may look very different compared to mine, so do not use my results for your lab. You're going to zoom in on these test tubes, and all you have to do is make an observation. You're not actually going to be doing anything with the test tubes. I'm sorry, I'm trying to get it. I'm sorry to make you dizzy. All right, so you're just going to observe. You don't have to remove any stoppers. You don't have to test anything. You're just going to observe. Remember, this was the egg in water. This was our control. This is the egg with just pepsin. This is the egg with the acid, the hydrochloric acid. This is the egg with the base. This is the egg with the pepsin and base. And this is the egg with the pepsin and acid. And you have to determine what happened here. Was the egg digested or not? And what from your observation will tell you that? That's up to you. So on your chart, on your lab report, you're going to go down to the data table that says protein digestion data. You've already made your prediction, and you should already have your test strip results as far as pH goes. And you're going to answer yes or no as to whether or not the protein was digested for each one of these test tubes. Then you have to put those in order of the greatest amount of digestion to the least amount of digestion. And you have to use your brain for this one. It may not be visible in the lab. It may not be very clear through your observations. So use your brain for that one and think about how human digestion works. Finally, you're able to answer number 8 and number 9. So in number 8, you'll be able to compare the contents of the digestive results for the protein. So you're looking again at the egg test, tubes 2, 3, and 5, and you're going to describe the optimal chemical surroundings for pepsin to break down proteins. And then number 9 says you're going to explain how this activity, which test tube was most like human digestion. If you haven't already filled out number 1 and 2, 3, 4, 5, please do that. That's part of your lab report. Also, your hypothesis will count towards your lab report. Remember, this lab is worth 40 points, so please do a good job on this. And when you are done, turn it into the Dropbox in your class. If you have any questions, please let me know. And thanks for watching. Bye.